Proteins are the most biologically diverse group of organic molecules there are. They fulfill so many structural and functional roles in organisms, it makes them super important. As you can see here, this is an enzyme, and you can see it's an elaborate molecule full of complexity. So let's explore them a little further. So as I said, there are so many structural and functional roles, an exhaustive list would be extremely long, but we can highlight a few of these things now. An enzyme is important for facilitating chemical reactions, making or breaking molecules. And we'll talk more about enzymes in another video, but they are a group of proteins. Certain proteins also transport other molecules and ions, like hemoglobin, transporting oxygen through your blood. Proteins can strengthen structures like your hair. The protein keratin is important in hair, and tissues. The protein collagen is important for most of our biological tissues. Muscle contractions are the interactions of two proteins, actin and myosin. And there are also proteins that play roles as hormones, transmitting chemical messages like insulin, which lowers your glucose levels in your blood. Now, the repeating subunits of proteins, or the monomers of proteins, are amino acids, pictured here. Now, all amino acids have these following parts: a central carbon atom, an amino group, which is an NH2 group, a carboxyl group, you can see there, and then finally this functional group designated as with an R that you see below the central carbon. That R group can be anything from a very simple group of atoms to a very complex molecule. And in fact, in proteins there are 20 different functional groups, which means that there are 20 different amino acids. So let's take a look at one of them, alanine. First, we notice the central carbon, the amino group, and the carboxyl group. So this CH3 group here is the R group that makes alanine, specifically alanine. And we compare it to tryptophan and glutamine, and we see their R groups are very different. And you may ask yourself, are proteins hydrophobic or are they hydrophilic? And that's going to depend on the identity of that R group. Some are polar, and some are nonpolar, and some even carry a charge. And those things are going to influence their interaction with water. So the repeated monomers amino acids are joined together by peptide bonds in proteins. So you can see this reaction here. Now, peptide bonds is just the name we give for the covalent bonds that form through a dehydration reaction in creating a protein. So you can see here that the OH group of a carboxyl group on one amino acid is interacting with the hydrogen of the amino group on the other amino acid. So they're outlined in red there and above. And then that is a dehydration reaction, so that OH group and hydrogen group leave as water, H2O, and a covalent bond is formed between the carbon and the nitrogen. You can see down in the lower left of that figure. And that covalent bond is just called a peptide bond when it forms between amino acids. So the terminology, just to get it straight here, peptides are what we call short chains of amino acids, and it's an arbitrary number, but typically less than 50. Polypeptides, then, are long chains of amino acids, typically over 50. Proteins are either one or more polypeptides joined together. Proteins have 4 levels of structure that are very important to their proper function. The primary structure, the first level, is just simply the sequence of amino acids in the chain. So as you can see here, just one amino acid after another in a certain sequence is the primary structure. The secondary structure of proteins arises when these chains of amino acids start to coil or form these flat sheets. As you can see here, the coiled regions are referred to as alpha helices, and the flat regions are called beta-pleated sheets. And this occurs when these peptide sequences start to interact with each other through hydrogen bonding, and that's what causes this coiling up or flattening out. So this picture is also representative of the third level of structure of proteins, the tertiary structure. This is where we get a further folding of the protein. So what you can see here is the secondary structures, the alpha helices, are forming almost right angles to each other in the protein, and that's giving it an overall shape. and that's because of further bonding between the different peptide regions. And then finally, the fourth and highest level of structure, the quaternary structure, again shown here by that enzyme we saw at the beginning of the video. This is when two or more of these polypeptide subunits are interacting. So here we can see 1, 2, 3, and 4 different polypeptides that have all come together and interacted in such a way as to give a large structure overall. Now the take-home message of all of these structural levels and all of this folding that we see is that a protein's shape has to be correct for it to function properly. And we're going to see that specifically with enzymes in the next video.